Hello everyone and welcome to our 15th online workshop, a focus on Canadian research. I'm Susie Gear, Chief Executive of Alport UK, a patient and volunteer, and I'm also a mother of 2 boys with Alport syndrome and have many other members of my family impacted by Alport syndrome. Let's introduce our 2 speakers from Canada. Firstly, Dr. Moumita Barua, an assistant professor of medicine at the University of Toronto and a nephrologist at Toronto General Hospital. And she will talk today about the studies that suggest that the frequency of variants in COL4A3 and COL4A4 associated with Alport syndrome phenotypes is much higher than historic prevalence reports. We first met Moumita at the American Society of Nephrology and were intrigued by her work to improve diagnosis, and so are looking forward to the talk today. Secondly, Vanessa Williams, a postdoctoral researcher at the University of Toronto, who will present her work in an Alport mouse model where she studied mechanisms underlying fibrosis and chronic kidney disease. And to date, we're very privileged to have 2 international scientific moderators. Ron Korstanje, an associate professor at the Jackson Laboratory in Bar Harbor, Maine, USA, and Rosa Torre, an adult nephrologist responsible for the Inherited Kidney Diseases Unit at Fundació Puigverd in Barcelona. And she's also professor at the Autonomous University of Barcelona. So Ron and Rosa will help me put questions to our speakers. Moumita, could you please share your screen with us?
Sure.
Great. We can see your slides, just need to be in presentation function.
Yeah, I think I have to swap my.
There, you've got it. That's good.
Okay, well, thank you. Thanks so much for that kind introduction, and great to be with you all again. I did talk at this forum about close to a year ago now in March, and I was delighted to get another invitation. So the title of my presentation today is Type 4 collagen gene variants lead to hematuria and albuminuria in UK biobank participants. Now, I think for many of you, these terms will be unfamiliar, but hopefully we'll work through what each of these mean in the next 20 minutes or a little bit less than that, I think. But broadly, I'm going to talk about 2 concepts. So first, I'm going to talk about families with Alport syndrome and the genetic characteristics that influence disease severity. And then secondly, I'll talk about the UK Biobank in particular. This is a database containing the genetic and clinical information of approximately 500,000 UK adult volunteers that represent the general population. And from this database has emerged several studies that have identified type 4 collagen gene variants in adults with hematuria and albuminuria, which are signs and symptoms that are seen in Alport syndrome. So I know this is well known to the majority, if not all, of this audience, but the classic form of Alport syndrome was described by Dr. Cecil Alport in the 1950s. And it was characterized at that time by a triad of symptoms consisting of kidney disease, eye deformities, and sensory neural hearing loss. And in particular, the kidney disease manifested as blood in the urine or albumin protein in the urine. So blood in the urine is known as hematuria, albumin protein in the urine is known as albuminuria, and progressed to kidney failure. So Alport syndrome is a genetic disorder that arises from mutations, what we now call pathogenic variants, in the type 4 collagen genes. And since its original description, we've actually gained a deeper understanding of the variability of symptoms seen in Alport syndrome. There are individuals who have mild disease characterized by hematuria, a low amount of albuminuria, without progressive renal disease and no detectable hearing or eye abnormalities. Then there are people on the opposite end of the spectrum who have that classic form of disease that was described by Dr. Alport, with the triad of symptoms involving the kidneys, leading to kidney failure, eye and ear abnormalities. And so the question is, what influences this variability? And to try to answer this, we have to first understand some terminology. I'm sure everyone has heard the term DNA, but might not have an understanding of what that is exactly. So DNA is packaged into units called chromosomes, and we have 22 pairs of chromosomes, numbered 1 to 22, one set from our mom and one set from our dad, for a total of 44 chromosomes, which are known as autosomes. Additionally, we each have a pair of sex chromosomes. Females have XX and males have XY. Each chromosome is then further divided into subunits called genes. We therefore have also 2 copies of each gene, one from mom and one from dad. And genes ultimately encode proteins, which comprise the functional machinery of a cell. So now let's try to understand the word gene variant. Actually, it's recently come into our terminology because we read about COVID variants, and they are kind of related. Each gene has a letter code, and here I show a cartoon of a 10-letter sequence in 3 individuals. 2 individuals are of European ancestry and one is of African descent. Oops, sorry. And we see that there are many similarities between their gene sequence, but we see that there are also differences. And there are more differences between individuals of different ancestral background compared to individuals of the same ancestral background. And these differences are what we call gene variants. Now, most of these gene variants actually define or dictate our traits, why I have black hair versus someone who has blonde hair. Or some of these variants are seemingly neutral, don't seem to have an effect on our phenotype or our characteristics or features. But a minority of these variants are associated with disease, and that is what we call a pathogenic variant. So pathogenic meaning disease causing. This previously was known as a mutation. And so Alport syndrome is as a result of pathogenic variants in the collagen 4-alpha-3, 4-alpha-4, or 4-alpha-5 genes. These genes encode the alpha-3, alpha-4, and alpha-5 protein subunits of type 4 collagen. And these protein subunits, or protein subunits, I should say, wind together like a braid, forming what we call a heterotrimer, and compose important structural elements of the kidney, eye, and ear, known as the basement membranes. So with that brief terminology description, let's talk about the genetic factors that influences severity in Alport syndrome families. So one factor is the number of copies of a pathogenic variant that an individual has. And in this cartoon, I depict a pathogenic variant as orange. Now, the collagen 4-alpha-5 gene is on the X chromosome and leads to what is called X-linked Alport syndrome. And in this form of Alport syndrome, males are more severely affected than females. The reason for that is this. So females have 2 X chromosomes, and so they have 2 collagen 4-alpha-5 genes. A female who inherits one pathogenic variant will have the other collagen 4-alpha-5 gene to compensate on the other X chromosome. By contrast, males have only one X chromosome and only one copy of the collagen 4-alpha-5 mutation, or pathogenic variant, I should say. And so they don't have another collagen 4-alpha-5 gene or on another X chromosome to compensate, and therefore they develop more severe disease. So a similar principle applies to the collagen 4-alpha-3 and collagen 4-alpha-4 genes, but these are not on the sex chromosomes. These are on the autosomes. They are both on chromosome 2. And so if someone inherits one copy of a pathogenic variant in either collagen 4-alpha-3 or 4-alpha-4, they will have more mild disease compared to someone who inherits 2 copies of a pathogenic variant. And one caveat that I will say is that even though I use the word milder, milder does not necessarily mean that the disease is not progressive. It can progress, but it might progress at a later age. For instance, the age of kidney failure might be later. Another characteristic that influences disease severity is the effect that the pathogenic variant has on the encoded protein. In a previous slide, I told you that that's what genes do. They encode for protein. So first, what I show you here is a gene without a pathogenic variant that we called wild type that encodes for normal collagen protein and therefore does not lead to disease. So secondly, I show you here a pathogenic variant known as a deletion that actually deletes part of the collagen protein and leads to severe disease. Similarly, there are pathogenic variants known as nonsense variants, which affect the encoded protein by leading to premature termination, or the protein is very short and non-functional. And this, like a deletion, leads to severe disease. And finally, I show you that there's a pathogenic variant known as a missense variant. This type of variant leads to the full-length protein, in this case collagen protein, being produced. But there is substitution of one of the building blocks, and this is a relatively mild change and therefore leads to milder disease. This is a figure that I've taken from a review paper that I co-authored with my colleague and mentor, Andrew Patterson, who's a panelist today, and it's currently in press in pediatric nephrology. So let's talk about the second concept that I want to cover today, and that is the findings from UK Biobank studies that have discovered or that have reported type 4 collagen variants in adults with hematuria and albuminuria. As I mentioned, the UK Biobank recruited approximately 500 adults aged 40 to 69 years, representing the so-called general population. I say the so-called general population because this population is actually a bit healthier and wealthier than the general population. Healthier because they excluded individuals with pediatric disorders. So all participants provided DNA for genetic characterization, as well as permission to access their clinical data that was generated during routine visits with their family doctors or specialists if they had any. In addition, a subset of individuals had tests that were specifically arranged by the UK Biobank. For instance, a subset of individuals had hearing and eyesight measurements. And so the purpose of the UK Biobank is to correlate genetic variation with clinical characteristics. What Dr. Patterson and I did is that we went onto the UK Biobank website last year, for instance, and we looked for clinical signs that could be seen in Alport syndrome and display our results in this table. When we accessed this last year, for instance, we found that 1,348 individuals had documented or persistent hematuria. About 420 individuals had a disorder of the lens that can also be seen in Alport syndrome, and 7,304 individuals had some form of hearing loss, either conductive or sensory neural. This is a screenshot from the UK Biobank that shows that a little over 200,000 participants completed hearing tests for the UK Biobank, which is available for approved researchers to access. And additionally, over 100,000 participants had eye measurements that is also available for approved researchers to access. And so the UK Biobank represents a powerful database where approved researchers can access it and look at their clinical outcome of interest. So for instance, a cardiologist might pull out everyone who has heart disease and try to find causal gene variants. There have been at least 5 groups that have accessed the UK Biobank data to correlate gene variants with urinary biomarkers, including hematuria and albuminuria, and I show their reports on this slide here. These groups looked at genetic variants that correlated with hematuria and albuminuria, which, as I mentioned, are clinical characteristics also observed in Alport syndrome. And interestingly, 3 of these 5 groups identified collagen 4 alpha 4 variants in UK Biobank participants with hematuria and albuminuria. So we show the actual collagen 4 alpha 4 variant information from these studies in this table, and this table also appears in our paper that I referred to. So 3 groups Haas et al., Sinnott Armstrong et al., and Talion et al. Reported collagen 4 alpha 4 variants in albuminuria and hematuria. And two of these groups, Sinnott Armstrong et al. And Talion et al., actually identified the exact same variant, the serine 969X nonsense variant. One group, Haas et al., identified a variant in the intron of collagen 4 alpha 4. The intron part of a gene is the part of the gene that does not code for protein. So you might ask, why didn't all three groups find the exact same finding? And if they're using the exact same database, they're all using the UK Biobank data. And so the reason for this is because they each use slightly different methodologies and cutoffs. But their associations are similar in terms of the variant or the signal allele, what we call allele frequency, p-value of association, and odds ratio. And so what we wonder is there actually really just one signal that they are finding, but because of differences in methodology, might have found something that's tagging that one underlying signal that's actually driving the association. And what we think that signal is the serine 969X variant. Now, focusing a little bit more on this variant, serine 969X has a frequency in the population, or I should say an allele frequency, which to make it easier for this audience, we'll call population, has an allele frequency of around 5 to 7 times 10 to the minus 4. And when we calculate that into a fraction, that actually translates into about 1 in 800 people will carry this variant. So about 1 in 800 individuals will carry the serine 969X variant. I can say people because most people carry only one copy of it. So for the first time, this gives us an estimate of the frequency of type 4 collagen variants leading to Alport syndrome symptoms in the population. And I would say that this is a minimum estimate. 1 in 800 individuals carries this variant that is associated with hematuria and albuminuria, but that is a minimum estimate because it only considers this one variant. And we know that there will be other variants in the population that will also be leading to hematuria and albuminuria. When we look at the literature, there has been estimates historically of Alport syndrome, and historic estimates have been about 1 in 50,000. This estimate is generated based on looking at the number of families and affected relatives with Alport syndrome and looking at the population number. But this historic estimate is quite biased because it only includes families with the most classic forms of the disease that are brought to specialist attention. It ignores individuals with milder disease, hematuria and albuminuria that might not progress and be brought to specialized attention and or assigned a diagnosis of Alport syndrome. So studies from the UK Biobank, which genetically characterizes adults that represent the so-called general population, suggest that type 4 collagen variants that lead to hematuria and albuminuria is at least 1 in 800, which is far more frequent than this historic estimate. So we were interested in this serine 969X variant in the UK Biobank as a result. And when we look at this variant in particular in the UK Biobank, we find that most individuals that have this variant have only one copy of the variant. 2 individuals had 2 copies of the variant, but majority had 1 copy of the variant. And this could explain why these individuals had milder disease in the way of hematuria and albuminuria. And so we looked further, we dug a little bit deeper into this serine 969X variant, and we find that in the Alport's literature, it is described as the commonest pathogenic variant in autosomal recessive Alport syndrome. And when we look at the family description, or the families that have this variant, 2 copies of this variant, we see that they have very classic forms of the disease characterized as Dr. Alport characterized it, with kidney failure and hearing abnormalities. And so taken together, this suggests that this variant has additive inheritance patterns, meaning that if you have one copy of the variant, you have mild disease, whereas two copies leads to more classic Alport syndrome. There are other genetically characterized cohorts like the UK Biobank, and one of those cohorts is called DECODE. DECODE is a genetically characterized cohort out of Iceland. And there is one study from DECODE that identified 3 variants in the collagen 4 alpha 3 gene as associated with hematuria. So to summarize, studies using UK Biobank and DECODE datasets have identified collagen 4 alpha 4 and collagen 4 alpha 3 variants in hematuria and albuminuria. And we show the location of these variants as a cartoon in this figure, which is also in our review paper. And so I'm going to conclude by these main messages. One is that population genetic studies reveal a strong association of collagen 4 alpha 3 and collagen 4 alpha 4 variants with hematuria and albuminuria. And we hypothesize that this serine 969X nonsense variant is driving the signal in UK Biobank studies. We also, the literature also suggests that the serine 969X nonsense variant follows an additive inheritance pattern, and that the frequency of variants in collagen 4 alpha 3 and 4 alpha 4, when we look at the frequency of those variants associated with hematuria and albuminuria together, suggests a higher prevalence, about 5 to 16 times higher than what has been reported previously. And that brings me to the conclusion of my presentation. I have to thank Dr. Patterson, who is online with us and really was the driving force of our review paper. And we are co-PIs on a project funded by the Alport Syndrome Foundation this year. And of course, also have to thank the efforts of the UK Biobank, participants, and the investigators that have done studies so far. Thank you.
Excellent. Moumita, thank you very much. It's clearly a really, really valuable resource to work on. While we're just about to ask you the first question, Harriet, can you get Andrew Patterson, Moumita's co-investigator on this project? He's going to join us as a panelist, so he can help answer some questions. Yes. Andrew is a professor in the Division of Epidemiology and Biostatistics at the Dalla Lana School of Public Health in Toronto, Canada. He's also involved in the program in genetics and genomic biology at the Hospital for Sick Children, Toronto, Canada as well. So welcome, Andrew.
Good evening or good afternoon, everyone.
Thank you. Okay, so my first question really for you before I hand over to Rosa is really to ask for some advice as a national patient group. When you're looking at figures like that and the kind of frequency of Alport syndrome, it's a no-brainer that people are just not being diagnosed. How can we improve diagnosis? How can we educate the professionals around the world to help us diagnose these cases so that people can access treatment and support? What do you recommend?
I think, so that's a great question. I think that's something that has been a focus for the Alport's community. For instance, for female, quote, carriers, heterozygous females with collagen 4 alpha 5 pathogenic variants. And so I think the efforts in that regard could apply broadly, that if someone has hematuria and albuminuria, they should be monitored. You should not just, if you don't have a reason for the hematuria and albuminuria, such as stones, kidney stones, you should be monitored. You should get annual blood and urine tests. And, but whether or not that would be enough to pique someone to have genetic testing, I think we're not at the point where everyone in the population is getting genetic testing. And so knowing if that's related to collagen gene variant, it could very well not be as well, hematuria and albuminuria in itself. So, In that case, so I think people should be monitored in terms of genetic testing right now, since it's not broadly for everyone. I would say if there is suggestion that the disease is progressing, if there's a family history of renal disease, with progression, if there is associated symptoms like hearing and eye abnormalities, then that should spark the thought of doing genetic testing.
Okay, excellent. Thank you. Right, over to Rosa first and then Ron to ask some questions.
Thank you, Moumita, for this excellent talk. And if I may, answering your question or adding something, I would add that when there's women that have hematuria, I would order a genetic test to rule out X-linked Alport syndrome, even if there's just hematuria. And of course, if the resources were unlimited, I would order genetic testing for
everyone.
For everyone.
Yeah.
But for adults, what I suggest nowadays is when you have a patient with proteinuria and hematuria, we always biopsy them. And if the renal biopsy is not diagnostic, for example, you find FSGS, which is the normal finding in advanced Alport syndrome, or whatever, anything that you cannot attribute an immunological cause, which is what you expect from a glomerular disease, then I suggest ordering a genetic test. Because patients may benefit from trials or from specific treatment in a few years' time. So I think they should.
Yeah, I agree with you. I think, though, sometimes it's hard to apply things broadly, and sometimes it depends on the individual patient. So I struggle sometimes to answer questions like this, but I agree with you. I think we don't biopsy enough, and at least in Canada.
I liked very much the numbers you showed, because I keep saying that I think that Alport is the most frequent genetic kidney disease, because and I base that in the paper from the group in Columbia, Grumman, published in the New England Journal of Medicine, where they found that the second most frequent monogenic cause of chronic kidney disease was Alport, collagen-4 genes mutations, and very, very close to polycystic kidney disease. But this cohort, it had all patients had chronic kidney disease. So if you add to that people with just micromaturia or micromaturia and microalbuminuria or even proteinuria, but without chronic kidney disease, that would increase amazingly the number. So I think it's probably most frequent, but there's something to be mentioned. Even being that frequent, which will lose the rare disease condition, but many of these patients never reach end-stage kidney disease. So I don't know how to do it, but somehow we should differentiate the subgroup of patients that may reach end-stage kidney disease to that enormous part that will never reach. So I don't know how this is seen from the rare disease condition, but it's different, for example, from polycystic kidney disease. They say it's not a rare disease because of its prevalence, but they mostly end up on dialysis, and it's not the case for this condition. So.
I mean, in terms of prediction, who progresses, so there are some of the factors that I described, but everyone's genetic background is different. And so they might have other risk variants, for instance. And that's why people are now, and have been for a while, talking about polygenic risk scores, where they find different, they found thousands of loci that are associated with increasing the risk, a little bit of for albuminuria or hematuria or reduced EGFR. And so taking someone's entire genetic makeup, not just one gene, not just like the collagen gene, but also accounting for the rest of their genetic makeup, probably will also be needed to predict and prognosticate.
Excellent. Should we hand over? Ron, do you want to ask a question? I noticed there are a couple of questions in the chat as well, but Ron, do you want to ask something?
Yeah, so I was actually stunned by the frequency 1 in 800, and this is only for one variant in one of the 3 collagen genes. So do you have any sense of, like, if in the UK Biobank you would sequence all 3 collagen genes, right? What would the frequency be of kind of like Alport mutations in the whole population?
So in the UK Biobank, these 5 studies, they've reported, I think those 2 variants, like that intronic variant and the S969X variant. And so the intronic variant we think is probably, and Andrew, you can like jump in anytime and correct me, but we postulate that it's tagging that one variant. And so like that's why, because they used a minor allele frequency that excluded that meant that they dropped the S969X variant. So from the UK Biobank studies, it seems like that's the only one that's associated with, that's the only collagen variant that's associated with hematuria and albuminuria. But then there's the DECODE one where they looked at hematuria, they didn't look at albuminuria. Maybe they looked at albuminuria, but they didn't find a signal. I can't remember actually. But when they looked at hematuria, they found these 3 variants in the collagen 4 alpha 3 gene. So if we account for the minor allele frequencies of all 3 variants plus the S969X variant, I don't think we did that. I can't remember if it's like what the denominator is, but we calculated that it was 5 to 16 times higher than 1 in 50,000. I'm sorry, I can't give you the denominator because I can't remember. Maybe Andrew, I don't know, Andrew, if you're able to answer that better than I just did.
I mean, I think it would be useful to discuss some of the history of this 969 variant. So as I remember, and Moumita can correct me, it was identified probably nearly 20 years ago in families with a classic Alport syndrome that were studied from the UK. So from my perspective, it appears to be what we would call a founder mutation, which is a genetic variation that is limited to a particular ethnic or geographic group of people. And at least from my recollection of reading those old papers that had studied classic Alport syndrome families, it was actually the most common 4A3 or 4A4 variant found in any of the British Alport syndrome patients. Is that correct, Moumita?
Yeah, yeah. That was from Judy Savage's Jason paper.
And I think in that original description, there were multiple families with that same mutation, which makes us think that it has a single origin in the evolution of humans and probably occurred at some point either before the British Isles were settled or relatively soon afterwards, and has kind of ended up being dispersed across the British Isles and is, as far as I can understand, currently thought to be the major cause of Alport syndrome due to 4A3 or 4A4 in the UK. Yeah. There is additional data that will become available, so whole exome sequencing and whole genome sequencing of the UK Biobank, which will allow us to examine all of the potential variants in all of these 3 genes to see their relative contributions to these phenotypes.
Wow, that sounds an even more expansive resource. That's incredible. It's really exciting work, and we really appreciate presenting it. I think in the interest of time, we may come back to you with additional questions, but if we get on with Vanessa's talk now, if that's possible. Vanessa, could you possibly share your screen, please? And I asked Rosa and Ron to have a look at the questions in the chat and perhaps ask those after Vanessa's talk, a couple of questions. It would be really good to bring out. Excellent, we can see your screen. Go ahead.
Great.
Can hear you well.
So thank you. Perfect. So thank you for having me, and it's a pleasure to share some of the work I did during my PhD in Dr. Jim Scully's lab. And my talk's entitled Exploring New Uses for Existing Therapeutics. And I'll go through how I use drug repurposing to identify treatments in an Alport mouse model. Okay, there we go. So treatment approaches that slow the progression of kidney disease are limited, but it's not for lack of trying. There are many reports of therapeutic candidates identified from studies in animal models, but they often fail during human trials. So the whole process takes roughly 15 years to complete at a per drug cost of 2.6 billion. And in rare diseases, such as Alport syndrome, the infrequency of disease adds additional barriers. Therefore, one of my overall research goals was to identify and test new treatment approaches to the progression of kidney disease in an Alport mouse model. And to do this, I use drug repurposing, which is an approach that may help to overcome barriers and facilitate drug discovery. When used in concert with more traditional approaches, it assigns new uses to drugs outside of the original scope, and therefore it can offer some key advantages like lower risk of failure since the drug has already been found to be sufficiently safe in humans, and reduce time to market. It can also uncover new information about the processes responsible for disease progression. Historically, most examples of successfully repurposed drugs involve serendipity or having some specific knowledge about how the drugs work already, and potential diseases that might respond well to the drug. However, with more recent advances in technology, computing, and our ability to acquire vast amounts of data, more systematic drug repurposing approaches are possible, and these approaches are becoming more and more popular. So for the rest of my talk today, I'll show you how we use drug repurposing to identify and then test new treatments. The first approach was based on specific knowledge about disease progression in the mouse. And then the second approach was a computer-based strategy. Then I'll briefly talk about some possible future avenues. So on to the first approach. This approach was based on knowledge that blockade of the Renin-Angiotensin system with ACE inhibitors, for example, is the current frontline of kidney disease treatment. And in both mice and humans, these drugs preserve function and reduce proteinuria, the presence of protein in the urine. And they have been shown to prolong the lifespan of Alport mice and delay the risk for progression in children. And so our lab wanted to more carefully study the components of the Renin-Angiotensin system in the kidneys of the Alport mice. So here are components of the Renin-Angiotensin system in which a series of steps leads to the generation of a protein called angiotensin 2 or Ang2 by ACE. And then Ang2 then interacts with another protein, its receptor, and this interaction leads to activations of processes that contribute to the worsening of kidney disease. And so ACE inhibitor drugs block the ability of ACE to generate Ang2. And so our first major but somewhat unsurprising observation was that Ang2 levels were increased in Alport mouse kidneys compared to the healthy wild-type control kidneys. And then the story gets a little bit more complicated in that another protein called ACE2 effectively lowers the levels of Ang2 by converting Ang2 into another protein called Ang17. And Ang17 also interacts with the receptor, which counterbalances the actions of Ang2, and this plays more of a protective role in the kidney. And so our second major observation was that ACE2 was decreased in Alport mouse kidneys. So we thought that if we could treat the mice with ACE2, it might lower Ang2 and increase Ang17, and then in effect contribute to slowing the progression of kidney disease. So shown here on the slide is a timeline of the Alport mouse lifespan in weeks of age. And I used a mouse model that has a variant or a mutation in the collagen type 4 alpha 3 gene. And of course, collagen 4 alpha 3 is one of the important components for the structure of the glomerular basement membrane, part of the blood filtering part of the kidney. And these mice do not produce a stable functional COL4 alpha 3 protein and mimic many of the features in humans, but on a much more condensed and accelerated timeline. So I studied 3 groups of mice, a group of wild-type mice with normally functioning collagen 4 alpha 3, a group of Alport mice that received placebo, and a third group of mice, a group of Alport mice that received ACE2. And then I began treatment at 4 weeks of age when there's quite mild injury, and then continued until 7 weeks of age, at which the point the disease has progressively worsened, approaching end-stage kidney disease. And then I assessed the kidneys. So here are some of the results. Shown here are images of the kidney under the microscope that we assessed for injury. What's notable, though, is the data shown in the graphs below. First, on the left-hand side, the graph shows us measurement of the protein albumin in the urine and is an indicator of the kidney filter function. And the order and colors of the bars correspond with the groups. So white represents the wild-type control mice, gray represents the Alport mice that received the placebo, and then blue represents the mice that received ACE2. And treatment was able to reduce the levels of albumin in the urine, signaling that treatment was able to preserve some function. And then on the right-hand side, we see a measurement of a protein called NGAL in the urine, and this gives us an indication of the health of the kidney tubules, which are responsible for reabsorbing water, electrolytes, and removing waste. And again, ACE2 treatment was able to lower levels, indicating that the treatment may have prevented damage to the tubules. And then as the disease worsens, fibrosis occurs, and during fibrosis, the normally functioning kidney tissue becomes replaced by scar tissue, and then it's not able to function sufficiently. And so I also examined the effect of treatment on this process. The images are representative of fibrosis under the microscope, and then the graphs show measurements of different factors that are known to be involved in and promote fibrosis. And so across the board, ACE2 treatment tended to lower levels of these factors. So to summarize, treatment with ACE2 lessens kidney injury and fibrosis in Alport mice. So now I'll talk a bit about the second approach, which involves using computers to identify treatments. So here's a workflow that was used. And here I studied groups of Alport mice as well as wild-type control mice at both 4 and 7 weeks from early to more advanced disease. A portion of the kidney was taken to simultaneously measure the gene expression or gene activity of about 30,000 genes in the kidney tissue. And then from that data, we created a profile, a gene profile of characteristics of the changes within the kidney in the Alport mouse kidneys over time in comparison to the wild-type kidneys. And we call this the disease progression signature. Then the progression signature was used to query a database called the connectivity map. And this database allowed us to screen about 1,000 drugs very quickly and predict which ones would most likely reverse the patterns observed in the Alport mouse kidneys. And many of these drugs in the connectivity map already have preclinical and clinical trial data, and they have established safety profiles in humans. And the top candidate drug predicted to reverse the gene expression profile characteristic in the Alport mouse kidneys was Brainostat. So what is Brainostat? Well, it's a drug that blocks a group of proteins called lysine deacetylases, and this can alter gene expression as well as the stability and activity of some proteins. But importantly, these effects mean that Brainostat can impact regulation of various processes and pathways known to be involved in kidney disease progression. So following a similar design that I used in approach 1, I studied 3 groups of mice, a group of wild-type mice, which will be represented by white dots in the upcoming graphs, a group of Alport mice that received placebo represented in gray, and then a group of Alport mice that received Brainostat, which would be represented in yellow. And I studied them again from 4 to 7 weeks. The effect of Brainostat on glomerular injury and urine albumin was variable, but the treatment lowered the tubular injury marker. I also examined the effect of Brainostat on kidney fibrosis, and Brainostat treatment decreased fibrosis-related factors. And then finally, I assessed additional cohorts of Alport mice for the impact of Brainostat on survival. And although the difference was modest, the mice that were treated lived longer. So in summary, treatment with Brainostat reduces kidney injury and fibrosis in the Alport mice, and in addition, Brainostat prolongs their lives. So now that I've shared some data on these 2 treatments, I'm going to share a bit about future directions. So one of the attractive aspects of drug repurposing is the fact that clinical data often already exists. And in the case of both ACE2 and Brainostat, they have been tested in healthy human volunteer subjects or patients with diseases other than kidney disease. And then interrogating like the kidney specific measurements from these trial data sets could be a first step toward determining whether or not they're feasible. But of course, we would still need to determine some key information, like whether or not these drugs are safe in people with impaired kidney function, and of course, whether or not they actually do provide kidney protection in people. Other considerations could include like combination therapy, specifically combinations with ACE inhibitors since they're already used in clinic. For example, I know that another group has tried Brainostat in combination with ACE inhibitor in a different kidney disease mouse model. And then we actually have tried ACE inhibitor in combination with ACE2, and in both cases, the combinations appear to maybe be additive in the sense that the combinations appear to be more effective at reducing injury together in combination than each drug individually. And then another thing to consider is that the connectivity map has grown substantially since it was used for this project. When I used it, there were about 1,000 drugs, but now there are 5,000. So it might be interesting to see if different candidates emerge. And then my final point, trying to end on a positive note, is that as difficult as the pandemic has been, it has forced a wave of innovation and the emergence of new ways of developing therapeutics. And more importantly, I think it's caused a bit of a shift toward more interdisciplinary collaboration, knowledge and data sharing, which are areas that the Alport community has been ahead of the curve, I would say. And since the pandemic, we've seen a lot of unlikely collaborators and rivals teaming up. And so I think these shifts could benefit drug discovery in the long term. And so finally, I'd like to take the opportunity to recognize our funders and the people who have made contributions, especially my thesis supervisor, Drs. Bay and Fang for their work on the ACE2 project, and Drs. Convalinga and Song for their work on the connectivity map project. So thank you for your attention, and I'll be happy to take questions.
Excellent, Vanessa. It's really encouraging to actually hear that the pandemic is actually pushing the collaboration further. And as we've very much promoted through our international workshops, these collaborative aspects of the work that you do is just vital, and we couldn't encourage it more. And it's really exciting to hear you talking about that. I want to ask you a personal question as to what you're going to be doing next in terms of, are you staying on and doing more studies on this, I hope, she says?
So recently, I've been working on projects regarding ACE2, as it's been identified as the receptor for SARS-CoV-2. In the long term, I do hope to get into like the biotech and the pharmaceutical space, hoping that like once I get in, I can advocate for rare diseases because they're often underlooked. But that's what I have been doing so far of late, and what I hope to do in the future.
Oh, well, I wish you the best of luck. And if you need some recommendations, as patients, we're really keen to support people. And I really mean that. We're forever writing letters and things to support those kinds of connections. So please come back to us. But thank you very much. Over to Ron. Do you want to ask some questions?
Sure. So yeah, that was a great talk and very interesting. One thing I was wondering, as you start your treatment at 4 weeks of age, which in mice is very young, and seeing that often in human patients, right, the disease is identified at a much later age. Do you guys have plans to try to do this treatment, but then starting at a later age when there's maybe more disease progression, and see if it still has an effect? And then my second question is whether these studies were done in males and females, and whether you saw a difference.
Okay, so the first question was about whether or not we were thinking about starting later. And so, yeah, that question had crossed my mind, but I didn't try it at all. I understand the benefit of doing that, but it's not something that I tried or investigated. And then the second question was about whether or not we did it in females as well, and the answer to that is no, we didn't. So of course, resources permitted, we would want to do the whole experiment in females to see whether or not there are sex differences, but we just did not perform that experiment.
Okay.
There's a question for you, Vanessa, from Francisca Lausecker. How do these drugs compare to other ACE inhibitors like Ramipril? And there's a second question in the same one. Also, are there any insights on the mechanisms, how the second drug works and ameliorates disease progression in Alport syndrome?
Okay, so the first question, how do they compare to ACE inhibitors? So I did not compare them directly, although I did mention that we tried ACE2 in combination with Ramipril, actually specifically, and we only measured albuminuria, but haven't measured other markers. And the combination, as I mentioned, looks to be additive, so that the combination works better than ACE2 on its own. So the second question was about mechanisms of the second drug, Brainostat. I believe that's what the second question was. So I specifically looked at two potential mechanisms. So I looked at the effect of albumin stimulation of tubular cells, and then also the stimulation of tubular cells with Ang2, and to see whether or not Brainostat had an effect on the activation of activator protein 1, which is important for it activates inflammatory pathways, it activates fibrosis pathways, and we saw that in both cases, Brainostat was able to attenuate the activation of activator protein 1, and then also the upstream MAP kinases that often regulate activator protein 1. So that's one potential mechanism. I did not study others, but Brainostat, it's not really it's quite a promiscuous drug in the sense that its targets, which are lysine deacetylases, it targets 2 classes, and that can have a lot of effects in general. So it's likely that many things are happening, and it would be hard to specifically pinpoint one specific effect of Brainostat. So likely there are many pathways and processes that are affected by Brainostat treatment.
Thank you. Thank you very much.
Right. Okay. I'm just wondering if we've got any more questions. I think probably.
I don't see any.
No, I don't see any either. Okay. Great. We will move on. Okay, right. Well, I think probably it's important to pass on our thanks to everyone for coming today. And please do let us know if you have any topics or research that you would like us to feature in our future workshops. Workshops this year will feature the latest research from Jai Ding's lab in Beijing, China, where they're doing exciting clinical work on genetics and kidney function. We've got Laura Perrin's lab in Los Angeles on the west coast of the States, where the lab are working on stem cell therapy ideas. And then back to Europe in Alessandro Ranieri's lab in Siena, Italy, who are working on genetics, gene therapy, and COVID-specific research. And I think Moumita will be joining us back again in July to update us on her lab's research there. So we shall look forward to that too. But finally, a huge thank you to Moumita and Vanessa. Moumita, we wish you well with your ongoing research, and Vanessa, to you and to James Shirley's lab too. Thank you also to our wonderful expert moderators, Ron in the States and Rosa in Spain. We really appreciate you joining today. It's fantastic to ask different questions from different perspectives. Finally, a massive thank you to Kidney Research UK for your sponsorship, helping fund our Zoom subscription and keeping the lights on for all these Alport online workshops. If you have any more feedback about this session or want to be added to our mailing list, email research@alport.info. Patients can also join the Alport Warriors Facebook group, and anyone can hit us up on our Twitter page. That's @AlportUK. We will get a recording of this talk up on YouTube in a few days' time. Otherwise, a big thank you to all of you so much for watching, and have a lovely rest of the day or evening wherever you are. Thank you.
Thank you.